Nucleic acid. Nucleic acid are large biological molecules that store and transfer genetic information. They are two types: deoxyribonucleic acid, ribonucleic acid. Where are they found? DNA found mainly in the nucleus of cell, also found in mitochondria and chloroplast. RNA found mainly in cytoplasm, also present in the nucleus. Type of RNA: messenger RNA, mRNA, carries genetic messages from DNA to ribosome, tell the cell which protein to make. Ribosomal RNA, rRNA, form the structure of ribosome, help in protein synthesis. Transfer RNA বা tRNA carries amino acid to ribosome, help build protein. Functions of nucleic acid: DNA store hereditary information, passes genetic information from parents to offspring, controls cell activities. mRNA carries instruction for making protein. rRNA form ribosome, help protein synthesis. tRNA deliver amino acid to ribosome. Viral RNA: in many viruses, RNA act as the genetic material. What is nucleotide? A nucleotide is a nucleoside that form a phosphate ester with the C5OH group of a sugar. Adenylate guanylate, citidylate, ইউরিডাইলেট, এগুলো হচ্ছে নিউক্লিওটাইডের উদাহরণ। নিউক্লিওসাইড কি? বেস প্লাস সুগার। নিউক্লিওটাইড কি? বেস, নাইট্রোজেন বেস, পেন্টোজ সুগার আর ফসফেট। Functions of nucleotide. Nucleotide are the building block of DNA and RNA. Important function: building block, form DNA and RNA, component of coenzyme NAD, NADP, FAD, coenzyme A. Energy storage: ATP is the energy currency of the cell. Cell signaling: CAMP, cyclic AMP, CGMP, cyclic GMP. These act as second messenger in hormone action. Component of nucleic acid. Every nucleotide has three parts: PSB, nitrogenous base, pentose sugar, phosphate sugar. Nitrogenous base, purine, double ring, adenine, guanine, pyrimidine, single ring, সাইটোসিন, থায়ামিন, থায়ামিনটা ডিএনএতে শুধু থাকে, ইউরাসিল, ডিএনএ, সুগার, ডিএনএ, ডিঅক্সিরাইবোজ সুগার, আরএনএ, রাইবোজ সুগার, সাইটোসিন, থায়ামিন থাকে, আর আরএনএতে সাইটোসিন, ইউরাসিল থাকে পাইরিমিডিন হিসেবে। ফিচার: full name of DNA: deoxyribonucleic acid, RNA ribonucleic acid, sugar deoxyribose, RNA ribose, bases ATGC, RNA হচ্ছে AUGC, strand হচ্ছে ডাবল, RNA হচ্ছে ইউজুয়ালি সিঙ্গেল। ফাংশন: স্টোর জেনেটিক ইনফরমেশন, মোর স্টেবল। ফাংশন: প্রোটিন সিনথেসিস, লেস স্টেবল। Watson and Crick model of DNA, 1953. James Watson and Francis Crick proposed the double helix model of DNA, 1953. Main feature: double helix. DNA consists of two strands twisted around each other. Right-handed helix. The helix twists to the right. Anti-parallel strand. One strand run 5-3, other run 3-5. They run in opposite direction. Diameter: DNA width 20 angstrom, 2 nanometer. 1 turn of helix, 1 complete turn is 34 angstrom. Contain 10 base pairs. Distance between 2 base pair 3.4 angstrom. Complementary base pairing. Complementary base pairing: A double bond T, 2 hydrogen bond present. Complementary base pairing: A double bond T, 2 hydrogen bond present. Z triple bond 3, Z triple bond C, 3 hydrogen bond present. এটা stronger. Purine pairs with pyrimidine. A হলো purine, T হলো pyrimidine, G হলো purine, C হলো pyrimidine. This keep DNA weight constant. Charge rule in DNA: A double bond T, Z triple bond C. A plus G equal T plus C. Total purine equal total pyrimidine. DNA is the genetic material. Hershey Chase experiment 1952. Alfred Hershey and Martha Chase used bacteriophage viruses that infect bacteria. What they did: DNA was labeled with radioactive phosphorus P32. Protein coat was labeled with radioactive sulfur 35. Virus infected bacteria. New viruses contained radioactive DNA. Conclusion: DNA entered the bacterial cell and passes information to offspring. So we can say that DNA is the genetic material. Chargaff rule: DNA's base composition. Regarding Erwin-Chargaff rule, total purine is equal total pyrimidine. Physiochemical properties of DNA: UV light absorption. Purine and pyrimidine bases absorb ultraviolet light strongly. Maximum absorption occur around 260 nanometer. This property is used to measure DNA concentration. Alkali stability. DNA is stable in dilute alkali. RNA is unstable in alkali. RNA contain a 2.OH group on ribose sugar. This group attacked the phosphodiester bond and break RNA. RNA does not have this 2'OH group, therefore DNA is more stable. Both contains AGC nucleotide phosphodiester bond and both involved in protein synthesis. DNA melting: when DNA is heated, hydrogen bond break, the double helix open into two single strand. This process is called DNA melting. Denaturation: অতিরিক্ত তাপ দিয়ে ডিএনএ এর দুই সুতার মাঝের বন্ধন ভেঙে দেয়া বা আলাদা করে দেওয়া। melting temperature Tm, temperature at which 50% of the DNA becomes single stranded. DNA with higher Gc content has a higher Tm because Gc pair are more stable than At pair. Annealing. After melting, if temperature is lower, slowly the two DNA strands join together again. This process is called annealing. Renaturation. Hyperchromic effect. When DNA melt, more UV light is absorbed. This increase in absorbance is called hyperchromic effect. Melting of DNA causes increased denatured UV absorption at 216 nanometer. Single stranded DNA absorb more UV light at 260 nanometer than double stranded DNA due to the unstacking of bases. In normal double-stranded DNA, bases are stuck together. Because of this stacking, UV absorption decreases. This is called hypochromic effect. Central dogma of life. DNA to DNA replication. ডিএনএ থেকে ডিএনএ রেপ্লিকেশনের মাধ্যমে তৈরি হলো। ডিএনএ থেকে ট্রান্সক্রিপশনের মাধ্যমে আরএনএ তৈরি হয়। আরএনএ থেকে ট্রান্সলেশনের মাধ্যমে প্রোটিন তৈরি হয়। ডিএনএ স্টোরস ইনফরমেশন, আরএনএ ক্যারিজ ইনফরমেশন, প্রোটিন পারফর্ম সেলুলার ফাংশন, ডিএনএ মেক্স আরএনএ, আরএনএ মেক্স প্রোটিন, ডিএনএ RNA protein. Central dogma is the fundamental framework explaining the one-way flow of genetic information within a biological system, proposed by Francis Crick in 1957. DNA to RNA, RNA to protein. The biological information flow from DNA to RNA and from there to protein. This is the central dogma of life. The DNA control every function of the cell through protein synthesis. Nucleoside, pentose sugar, nitrogen base, adenine, guanine, cytosine, uracil, thymine, nucleotide, phosphoric acid, pentose sugar, nitrogen base. Adenine plus sugar, adenosine, adenine plus pentose sugar plus nitrogen base, adenosine monophosphate, guanine plus sugar, guanosine, guanine plus phosphate sugar plus phosphate plus pentose sugar plus nitrogen base. অ্যাডেনিনটা এটা নিজে কি নাইট্রোজেন বেস, এর সাথে পেন্টোজ সুগার, তার সাথে ফসফেট, মানে অ্যাডিনোসিন থেকে অ্যাডিনোসিন মনোফসফেট, গুয়ানোসিন থেকে গুয়ানোসিন মনোফসফেট। সাইটোসিন থেকে কি আসলো? সাইটিডিন। সাইটিডিন থেকে সাইটিডিন মনোফসফেট। ইউরাসিল থেকে ইউরিডিন, ইউরিডিন মনোফসফেট। থায়ামিন থেকে থায়ামিডিন, থায়ামিডিন মনোফসফেট। পিউরিন বেসে নামের শেষে সিন বসে। অ্যাডেনিন গুয়ানিন থেকে অ্যাডিনোসিন গুয়ানোসিন। পাইরিমিডিন বেসে নামের শেষে ডিন বসে। সাইটোসিন সাইটিডিন থায়ামিডিন ইউরিডিন। মনো একটি ফসফেট থাকলে, দুইটা থাকলে ডাইফসফেট, তিনটা থাকলে ট্রাইফসফেট। অ্যাডিনোসিন ট্রাইফসফেট এটিপি। ডিঅক্সি adenosine monophosphate DAMP. RNA contain a 2OH group on its ribose sugar. In alkaline condition, the group can perform a nucleophilic attack on the phosphorus atom, breaking the phosphodiester linkage and degrading the molecule. DNA lack this 2OH group. It has a 2H instead, making it resistant to the specific type of alkaline hydrolysis. So DNA is more stable repository of genetic information. Replication DNA থেকে DNA. The process where a DNA molecule makes a identical copy of itself occur in the nucleus in eukaryotic during S phase of the cell cycle. Enzyme DNA polymerase. Transcription DNA to RNA. The process of copying a specific segment of DNA, a gene, into messenger RNA mRNA occur in the nucleus. RNA polymerase enzyme is used. Translation RNA to protein. The process where the ribosome read the mRNA sequence and convert it into a chain of amino acid, a protein, occur in the cytoplasm at the ribosome. Exception to the central dogma. Reverse transcription RNA to DNA. Retroviruses like HIV use an enzyme called reverse transcriptase to copy RNA back into DNA. Prions infectious protein that can alter the structure of other protein, changing information without involving nucleic acid. Genetic code. Genetic code is the rule that tell how DNA make protein. 3 DNA অথবা RNA later make one code word. This is called a codon. One codon mean one amino acid. AUG mean methionine. There are 64 codon in total. 61 codon make amino acid. 3 codon are stop signal. Codon is on mRNA. It has 3 letters. Codon tell which amino acid to add. Anticodon matches the codon. It bring the correct amino acid. If codon is AUG, then anticodon is UAC.